Nice to meet you.
Sir, you can start.
Yes, ma'am. Yes. Good evening, my dear students. You are all warm welcome to the wonderful session of the biological classes 2023. This is Dr. N. Meenakshi Sundaram, MSc, MPhil, PhD, and BA, BH, NEET biology faculties. So, past years, we did 70 to 80 classes already we did, and the test portion also we did, isn't it? In these sessions, we are going to discuss the remaining part of the biotechnology. Already we know about what is my biotechnology, what are the components, what are the steps involved, all those things already we discussed. In these sessions, what are the things we are going to discuss, you don't know. Already we discussed the vector-less gene transfer, isn't it? Vector-less, without the involvement of the vectors. Okay. The another one is called as vector-mediated gene transfer. Today, we are going to discuss about the vector-mediated gene transfer. What is the meaning of vector-mediated gene transfer? Means in between the donor and the recipients, there is a mediator. That mediator acts as a, what is a vector? So that's why they have given as a name is called as vector-mediated gene transfer. So from this heading, we must know what is by cloning vector. Already we studied these things also. Already you know about. Already we studied these things. Cloning vectors. So which one is the cloning vectors? Already you know about the plasmid, cosmids, and that things also comes under the heading cloning vectors. One of the most important element of the cloning vector is a recombinant DNA. The recombinant DNA. So what is the meaning of recombinant DNA? Already we discussed it. What is that? A donor as well as a recipient genes are united together, form as a single genes. That gene is called as vector. That is also a recombinant DNA. Technology or in the gene cloning is the vector. This is very, very important. Without these things, there is no any gene transformation takes place. Okay. Once again, one of the most important elements in the recombinant DNA technology or gene cloning is the vector. The random DNA or cDNA, so otherwise called as what is a circular DNA or complementary DNA segments. A specific gene is linked into a small replicating DNA molecule called a vector. Okay, once again repeat for you. Random, you can choose any plant, animals, whatever it may be. It may be prokaryote, it may be eukaryote. Okay, so cDNA segment, a small piece. A specific gene is linked into a small replicating DNA molecule is called as vector to form a DNA that is called as recombinant DNA. That's already told you in the introduction part. I told you, isn't it? So the RNA molecule which can be propagated in a suitable host cell. This is very, very important. That is the mechanisms. Without host cell, it will not undergo the replication process. Mostly in laboratory aspects, E. coli is used as a host cell. To a large number, not in the E. coli, and also some of the bacteriophage is also there. Okay. There are different types of cloning vectors for use with the different types of host cell. This is the difference. It will not accept all the host. It will not accept all the vectors. Each one, it has a specific replication host and replicating vectors also. Suppose if we are doing an opposite manner, what happened? The proliferation as well as recombination process, the multiplication process, it will not happen properly. As a result, what happened? You will not get the proper result. That is very, very important. Once again, repeat for you. Vector-mediated gene transfer. In this category, cloning vector is very, very essential one. So one of the most important element in the recombinant DNA technology or in the gene cloning in the vectors, the random DNA or cDNA segments. A specific gene is linked to the small replicating DNA molecules called vector to form a recombinant DNA molecule which can be propagated in suitable host cell to large numbers. There are different types of cloning vectors for use with different types of host cell. Okay, this is very, very important. Without these two things, it will not proceed the any gene transformation. So go to the next slide, my dear students. See that one? The largest numbers exist for E. coli. I already told you which one is mostly they were used as the vector because E. coli is nothing but the one of the prokaryotic molecules. It has an extra chromosomal body and also possesses some specific characteristics in the form of genes. So they are all considered getting known as what is that? Vectors. Otherwise called as see that, and the best known of these the plasmid vector. So this is very well because already known the structures and sequence. What is the general sequences present in it? What is the genetic code present in it? That things already be well known. That's why it is called as mostly preferable. Okay, that's why they prefer. What is that? Scientists prefer E. coli. The best known of these is the plasmid vector. Okay, go to the next one. Besides, there are cosmid pages. It's another part, cosmids. Okay, and phage means. Phage means you already know a bit. Phage. Phages means bacteriophage. Means it has a specific character. Extra chromosomal body is there. That extra chromosomal body named as a phage mid. Mids. And then yeast. Yeast is also one of the fungi. Already studied that things also. It is a very, very important. Yeast. The unicellular fungi. Others are multicellular. Okay. So not only that, and also yeast, artificial chromosomes, transpons, bacteria artificial chromosome, etc. These are all having the what is the characteristics? What is which characteristics? Vector characteristics. Sometimes they may ask the question, which one of the following is not a vector? Like that, they may ask the question. Sometimes they may ask the question, which one is the known one? Okay, best known of the scientist. Okay, like that, they may ask the questions. So these are the very, very important, my dear students. The largest number of the exist for the Escherichia coli, mostly. Besides, there are cosmetic phages and phage meats and yeast artificial chromosome and transpons, bacteria artificial chromosome, etc. These are all also considered the heading is known as vector. Okay, so go to the next one is choice of vector. How it will be selected? Okay, the choice of vector is very, very important. It is a replication process. Suppose if it is selected, it's it's improper means what happened with the replication process, the origin, the ori, what happened? It will also suffer. Finally, your result will be negative manner. So that's why the choice of vector is very, very important process. First, the vector is autonomously replicating inside the cortical. This is a vector. There is a ability. There is a potentiality. What is that? Automatically replicating inside the cortical. Once it's entered, the vector is entered in the cortical. What happened? Automatically, it undergo the cell process. What is a replication process? That type of gene. What is that? We can choose it. We can select it. Okay. So then only our desired quality or characters will be obtained. Suppose the selection of vector is wrong, means your process also will not get properly. Already told you. Then the DNA molecule designed from a plasmid or phage DNA to carry a foreign DNA inside the host cell. Okay. A vector should have the following features. This is very, very manual. You can take an example. Any one of the vector, these are the characters should possess. Otherwise, it is not considered that it is a vector. That is very, very important. So in most containing a replicon undergoes the replication process or ori. Ori gene already studied. And the replication process started a particular position or location. That particular gene is called as ori gene. Otherwise called origin of replication started. Okay, initiation stage. So that enables it to replicate in the host cell. This is very, very important. That it must contain which one? The vector must containing a replicon. The replicon is also otherwise called as ori. Ori is nothing but the origin of replication process that enable it to the replicate in the host cells. Okay, already we saw it, these things also. Okay, see that. So what are the things we have to discuss here? No, I will show you in the diagrammatical way. Then only it will be better for your understanding. See this one. Already we saw it. So E. coli is nothing but a small bacteria, isn't it? E. coli is nothing but a small bacteria. The inner side, what happened? There is a extra chromosomal body is there that is considered as a plasmid. Okay, these plasmids. Plasmids. This is considered as extra chromosomal body present in the phage or bacteria or whatever it may be, that is considered as a plasmids. Okay, this is E. coli. This is a bacteria is called as E. coli. Is mostly they used as a which one, you know, vector in laboratory aspects. Okay, this is the E. coli structures. Okay, this part, you know, so you can remove it already, you know, but you have to rupture these things, you will get outside, isn't it? The structure will be look like this. Okay, this is considered as a plasmids. Here, the replication, the vector, it possesses what are the following characters? Now, first of all, the ori. This is very one. First one is it possesses the ori. What is the meaning of ori? So, ori is nothing but the origin, replication starting place. This is called as a, what is that? Ori. First one, ori gene present. Ori. Okay, ori gene. This is very, very important. The ori gene, it must possess. Okay, where there is a ori, what does that mean? The origin of the replication process, the replication starter. That means polyandromic. Already we studied in the last classes, isn't it? Last classes already we studied. There is a polyandromic gene, means suppose you rewind this circular structure, you will get the linear structure, isn't it? So at that time, what happened? You know, so look like this one, isn't it? Already we studied these things. So the structure will be like this. This is considered as a 5-dozen. This one is considered as a 3-dozen. Already we discussed. Okay, each one it has a specific complementary gene is the 3-dozen and 5-dozen. Here the 5-dozen automatically. reversibly, irreversible manner. Okay. Okay. 3 dash end. So the ori is any place with the help of the restriction endonuclease. Already we studied that things also. Mostly there is a place. Just imagine this is the place of ori. This one is called as origin. Okay, the ori. Okay. So where it occurs, already you know about the palindromic. Polyandromic. Polyandronic gene. Okay, polyandronic genes. So polyandronic gene is the place. It can be easily made a cut in this position with the help of the enzyme is called as molecular scissors. Already we discussed the resection endonucleases. After that, what happened? It will be split. This character must be possessed. Which one you know? Vector. This character, replication of the ori. So this is the character of first one. Already we discussed the ori gene. Okay, so see that one. See the vectors should be the following. It must contain a replication on ori gene that enable to replicate the host cell. Next one is, see that it should have a several marker gene. It should have several marker gene which help to differentiate the transformed cell from the non-transformed cells. Okay, already we saw it. Which one is marked one? Which one is we are introducing? We should note it down, isn't it? We cannot be able to identify which genes we were introduced. Which gene is already present in the host cell. We cannot be able to find out. That time they were used, which one is several marker. Already we saw it in the PCR, polymerase chain reaction during the gene DNA fingerprinting or gene printing, fingerprint. That time, what happened? No, they were used the marker. Okay, not only that, and also in the even separation of the molecule, in where electrophoresis we were used the some what is that markers. Transform cell from the non-transformed cell, and also to identify the transformed cell which contain recombinant DNA molecules. What is the usage of the marker? You know, which one? It has the recombinant genes. Which one is the recombinant gene? We cannot able to find out, isn't it? That is the reason. So that is that is indicated a gene for ampicillin and a tetracycline resistant gene. That is the reason. So why we are using this one? You know, suppose any one of the microorganisms they are having the a specific plasmid is there, and also having possessive characters also. That character is decided by the gene. That gene, it has a ability, the desired quality, superior quality. What is that? You know, ampicillin resistant gene or tetracycline resistant gene, heavily identified with the help of the marker only. Okay, without marker we cannot be able to find out which one is the ampicillin. We can wrongly we can collect the sample. Suppose ampicillin gene is also, instead of ampicillin we may collect the tetracycline resistant gene also sometimes. So to prevent these activities we may use the several marker genes. Okay, so what are the ampicillin and the tetracycline resistance? And then it should have a unique cleavage site within one of the marker gene. So the insertion of the foreign DNA into the marker gene lead to other inactivation and identification of the recombinant DNA molecules. Not only that, it should have a unique cleavage site. Cleavage site means we can easily cut. Okay, within one of the marker genes, so that insertion of the foreign DNA, insertion. Why we are cleaving? We are making a cleavage? No, for the insertion of the foreign DNA into the marker gene leads to the inactivation and identification of the recombinant DNA molecules. These are the qualities. Okay, very, very important things is that. Next one is, see that one. For the expression of cloned DNA, that vector DNA should contain suitable control elements such as promoter, already we studied it, promoter, terminator, and ribosome binding site. When you are studying that, where in the protein biosynthesis translation process, the mRNA comes out from the nucleus, and it's also attached with the ribosomal RNA and then tRNA molecules. tRNA completely, what happened? Collect the amino acids correspondingly matching to the mRNA molecule. As a result, what happened? Promoter, otherwise called as initiator. The process, leading process is there. And then finally, the terminator is there. So these are the process occurs during the translation or protein biosynthesis. Same process also occur where, in the cloning methodology also. Okay, here, what are the processes? The control elements such as the promoters. Promoter is one of the control element. It started and also prevention is also. The promoter is not there, what happened there? The initiation completely stopped. Okay, so and the terminators, after completion of the process, what are the required materials synthesized by the DNA molecule or any other vectors molecule? How will it will be stopped? With the help of the terminator, there what happened? AUG is a starting codon during the protein biosynthesis. But in this category, what happened? There is a promoter is there. Likewise, the terminator is also there. Not only that, and also ribosomes binding site also there. There are three things is very, very important, just like a translation process. Okay. Okay. And the next one, which size we should prefer? The vector should be which, okay, whether it may be more than that or less than that. See that one, preferable small in size. Normally, but the E. coli bacteria is a very minute microscopic unicellular prokaryote, isn't it? It's a prokaryotic nature. Within the host, there is a small size. Which one, the plasmids. Okay. So we have to prefer that much size of the, what is a vector? Then it will be matched easily. Suppose if it is not matched easily, means what happened? There is a wrong selection is there. Ideal vector should be less than or equal 10 kilobase. This is very, very important, my dears. When you are selecting the process or when you are selecting the vector size should be less than that of equal of the 10 kilobase. Suppose if it is more, what happened now? The process is very complicated. The small size, see that one? The small size is essential for easy introduction in the cell. This is the important part. By transformation, transduction, and the electrophoretions. Because we cannot be able to load in the gene gun. Okay, suppose if it is more than that one, the molecular weight will be very high, we cannot be able to load, and also we cannot be able to transform from the outside to inside. So the size should be a 10 kilobase, which is also very, very important in the NEET point of view. Don't forget, my dear students. The small size is essential for the easy introduction in all cell by transformation, transduction, and also the electrophoresis. These are the three methods. Okay, so don't forget. And go to the next one, the stability in host cell. And one more is that it's very, very important. What is my stability in host cell? When we are introducing the vector inside the host, it should be stable. Stable in nature. Suppose if it is not stable, suppose if it is outside stable, when you are introducing, after the introduction, what happened? The molecule, it will be unstable mode. What is that? It is not the vector characteristics. So you select the stability vectors. See that vectors should be stable in the host cell after introductions. This is very, very important. After introduction. And should not get, okay, should not get lost in the subsequent generations. This permits replication of the vectors producing large copy of gene of the interest. So this is the important point. The stability in the host cell is also very, very important. Okay. And then next one is the ability to control their own replication. It has the ability to control their own replication also. Okay, suppose if it is not uncontrolled, means what happened now? Once you are introducing inside, means it will not, after the completion process, it will not stop, means what happened now? It is very complicated. Suppose when you are introducing the other plants or animals, what happened, it will be very dangerous one. So that's why the ability to control their own replication also, that property is also very, very important for the vector. See, these properties enable them to multiply and exist in high copy number. Okay. See that. So what are the things in multiple cloning site? So one of the site is called as multiple cloning site. What is the meaning of MCO? Sometimes they may ask the question in the expansion. Which one of the following is just like a EDTA? Okay. And also a GIFT. Okay. And also ICBN, ICJETN. Other things also there. Very, very important. SDS. Okay. Sodium todicylsulfate. Like that. The MCO. Okay. Multiple cloning site. Okay. This property permits the insertion of gene interested and plasmid recirculations. Okay. This is the another things. What the next one is, should not be transferred by conjugation. Okay, should not be transferred by conjugation. The property of vector molecule prevent recombinant DNA to escape to natural population of bacteria. Okay, suppose E. coli is also one of the bacteria, isn't it? During the conjugation process, what happened? The replication process takes place. It is naturally occurring, means what happened, there is no need of which one, transformation. So it should not be transferred by conjugation. Such type of molecule is called as vector. This is the important property. Okay, so don't forget, my dear. So these are the very, very important things. Once again, repeat for you all those things. See here, what are the characteristics is present in choice of vector. You must note it down. The vector should have the following features: particular replication of the origin gene is also present. At the same time, what happened now? Once it transfer, means the non-transfer cells also identified with the help of the markers. Several markers we were used. And then next one is the preferably small in size. That means it should not be exceed 10 kb or equal to 10 kb only. Then only the transformation or also transaction and electrophoresis is takes place. Okay. And then the stability of the host cell is very, very important. Suppose if it is entered inside the host, it should be very stable in nature. Suppose if it is not very stable, means what happened, it is not a correct vector. And also the ability to control their own replication. This is also another property. Multiple cloning site is also there. That is also a very, very important for the recirculation process. And should not be transferred by conjugations. Suppose when the bacteria unexpectedly join together, as a result, what happened? The conjugation canal is also formed. During that time, what happened? The recombinant. It's possible, means what is that? It is not the gene transformation. We cannot be able to get the prior quality, superior quality. We will not get it. Automatically it happened means naturally it happened means what happened? We do not do this one, isn't it, in our lab? This next, very, very important, my dear students. Next one is very, very important. Examples of vectors. Now we have studied what is by vector-mediated gene transfer, and then what is by vectors. What are the general characteristic features of the vector? What are the general characteristics features of the vectors? Then, what are the examples of vectors? This is very, very important, my dear students. Examples of vectors, there are so many things, particularly the plasmids. It's a very, very important one. 100 times, 1000 times, note it down. This is very, very important. Okay, the plasmids already we discussed. Okay, the plasmids are double standard closed circular DNA. Note it down. A double standard closed DNA, circular DNA molecule which exists in the cell as an extra chromosomal unit. Don't forget, my dear. Plasmid, right? Plasmids are which standard? Sometimes they were asked the question. which forms double standard closed circular DNA molecule. Already we saw it in the see that. So this one, okay, this one is a double standard. This is a plasmid. See that structure is double standard. Okay, so this is considered a double standard. Double standard. Simply we may put steady standard. Okay, circular manner. See that circular manner. So it's also called as, what is that? Extra chromosomal body. Extra chromosomal body. Extra. So the DNA is also present inside, but other than the DNA molecules, that is extra chromosomal body. Chromosomal. Chromosomal body, extra chromosomal, double standard extra chromosomal body present inside the host. That is called as plasmid. Got it? So plasmid having these are the characters. Already we saw it, isn't it? That is very, very important. So, okay, go to the next one. And the plasmid replicate and are inherited independently. The plasmid range in size from about 1 kb to over 250 kb. We need only 10 kb only, isn't it? But here 1 to 250 kb. Sometimes they may ask the question, the size of the plasmid should be, which one of the following is the correct one? Sometimes they may ask the questions, wrong statement. Okay. Sometimes they ask the true or false. Sometimes they ask the question, direct questions. Once again, plasmid is very, very important. So we're taking first place. Double standard closed circular DNA molecule which exists in the cell as the extra chromosomal unit. Plasmid replicate and are inherited independently. Plasmid range. Okay. So range, which size, size from about 1 kb to over 250 kb. This is very, very important. Next. Based on the origin or source of the plasmid, they have been divided into two major classes. One is called as natural plasmids, another one is called as artificial plasmids. Natural means already will be any one of the microorganisms. Take an examples, bacteria. E. coli is one of the bacteria, it is a natural one, and also it has a additional characteristics. Additional characteristics. What is that? That is called as, what is that? Natural plasmids. The natural plasmids example that I have given in a E. coli, coli E1. Okay. Natural plasmid, they occur naturally in prokaryote or eukaryote. That is the reasons. Naturally present in the extra chromosomal body in the host cell. It can be isolated with the help of some special technique. Already know about. Okay. They occur naturally in prokaryote and eukaryote. Example, coli E1. Coli E1. Natural plasmid. Don't forget, my dear. This is very, very important. Not only that, and so many things are there. Second one is called as, what is that? Artificial plasmid. Artificial means man-made commercially or within the lab. See that one, how it will be. Comparison between the artificial as well as the natural plasmids. Naturally occurring plasmids has several limitations. For example, some are straightened and not relaxed. Some have fewer marker genes and some are too large. So these are the 3 points you have to remind. Some are straightened, okay, and are not relaxed. Some have poor marker genes. Poor marker genes. And the last one is some are too larger in size. So these are the differentiation of the natural as well as the artificial. Okay. To overcome the limitation of the natural vectors, artificial plasmid is designed by combining different elements from diverse sources. What is the meaning of diverse sources? Diverse sources means what is the different sources is called as diverse sources. We can take any one of the plasmids, the E. coli, some of them are isolated from the Bacteria, some of them are isolated from the plant. Likewise, they are all united together. You can construct your own gene. In a single gene, it contains different, what is a diverse sources. Diverse sources means one from, say, take an example. I will show you, then only it will be better. Okay, see that one. See here. Take an example. So this is considered a plasmid. We did not know any name. Okay. I saw your rough calculation. That one inside. Just imagine this one is enough. Okay. This is considered an extra chromosomal body, is called as E. coli. Here, the particular portion of this one, for example, this one, this portion. Okay. In this portion, named as A. Okay. Another session, another portion. Take an example in this portion. Okay, in this portion, take an example of B. And then, so in this portion, this consists as C. This one, T. Imagine T. Okay. And then another one, we can choose it another different colors. Okay, this one is in this portion. In this portion, you can to put D also. Okay. The last one, choose this part. F. So here we may give a different numbers, isn't it? Different alphabets, A, B, C, D, and F. Okay, and also T is there. This A gene is also the A in particular locations. Okay, the fragments of the each one, it has a specific fragment. Each fragment is also isolated from the different origin, different. Okay, so different origin. See that? So different means different elements or plants. Just take any examples. Different organism. You can imagine organisms. Okay, different organism. Imagine. For example, okay, different organisms. Imagine. So this part, take an example. EA. A is also isolated from the extra chromosomal body of the E. coli. Okay, E. coli part. This is taken examples of the plant. What is one, you know? Suppose taken examples of bacteria is also there. Taken examples of this one is called as plant. Any sources of the plant, tobacco plant or whatever it may, plant, PT cotton, whatever it may. C denoted as some other animals. Okay. For example, which one you know? That jellyfish. Okay. Jellyfish. Okay. Fish. And F is also another type. So D is also, T is also tetracycline. These are the, each one it has a specific property. Each one it has a specific antagonistic effect. Otherwise called as fight against many diseases, draft or cold or tetracycline, ampicillin, whatever it may be. Each one it has a specific characteristics it has. Okay. Suppose these are all are constructed in artificially. What happened? The particular genes, the plasmids, this is considered collectively called as plasmids. This is also commercially or laboratory we were made, commercially prepared. So this plasmid, it has different genes also, diverse genes. These are the diverse genes also isolated from the different organism and united together and form as a single plasmids. How it will be possible, you know all about the technology, molecular base of inheritance, we can cut any bases. That is called a stress section endonuclease under the ligation process. Here, just the E. coli bacteria is there. E. coli, that particular part is isolated from the E. coli bacteria and also joined together. So cutting and linking process also done by the specific molecular molecules. What are the molecules? You know, but there are two molecules is very, very important. One is, what is that? Cutting process. Okay. So cutting, what we are using? Okay. Which one is using? Cutting process. Okay. Sorry. Okay. Cutting process, what we are using? Already you know about the restriction endonuclease. Restriction. Okay. Restriction endonucleases. Okay. Nucleases. At the same time, second one, linking process, already you know about linking. Okay. So after the joining of more than one genes, these all are united with the help of linkage. With the help of the enzyme is called as, what is it, you know? That enzyme name is called as, what is that? Dhanalakshmi, do you know?
Sir?
DNA. Which enzyme is used for the linkage process?
Sir, don't know, sir.
You don't know what? Why? What about the two DNA molecule united with the help of the linking process? Which enzyme is used? DNA?
Ligase, sir. DNA ligase.
Yeah. That is very, very important. Okay. So these are the artificial MAD. Okay. So that is very, very important, my dear student. Don't forget. See, this is the important things. See this one. So diverse sources, they are constructed in vitro. That's why I told you the in vitro means within the lab by recombination, selected segment. This is very, very important. Selected segment means, that's why I told you, each one it has a specific characteristics of two or more other plasmids. Two or other plasmids, natural or artificial. For example, B plasmid BR322. Don't forget, my dear, this is very, very important. So many times they were asked the questions. This is very, very important. Okay. So the artificial plasmids, given examples. Which one is the artificial plasmids? BPR322. And also another one is also the BPR325. Okay. So this is very, very, this comes from where, you know, a selected segment of two or more other plasmids. That's why I told you the before slide we saw it, you know, that is the different sources obtained from the different organisms and also construct their single artificial plasmids within the lab in vitro nature. So finally they give you a name or gave you a name, is called as a BP plasmid BR322. Okay, so don't forget. See, that artificial plasmid vector was classified into two broad types based on their use. The artificial plasmids also further divided into two categories according to their usage. One is called as cloning vectors, another one is called as expression vectors. Now it will be, one is called as cloning vectors, another one is called as expression vector. Is the cloning vector is used for increasing the number of copies of your cloned DNA fragments. This is the important first point. And expression vector, it is used for the expression of foreign gene into the protein. So these are the two things with that. Okay, cloning vector and expression vector. Cloning vector, what they will do, you know, it may be increasing the number of copies. That is very, very important. During the cloning process, what happened? No, increase the number of copies. And another one is, what is that, you know, the expression vector. Expression vector, what happened? No, it express the gene into the protein molecule, because expression only we can see in the protein molecules only. These are the important things. Don't forget, my dears, once again. So the examples of the artificial vector or plasmids, already we discussed, BP or 322. We are going to study the structure. What are the characteristics features present in it? What are the gene present in it? What are the gene responsible for the production of antibiotics? That things we are going to see in the, okay. See that BP or 322. This was one of the first artificial cloning vector to be constructed. This is very, very important, my dear students. Sometimes they were asked the question. Last class, I think 3 or 4 question paper, just go and check it. Which one is artificial plasmids? Which one is not artificial plasmids? These two questions will be there. Check it. Okay. So first artificial cloning vector is name is called as BPR 322. Don't forget. To be constructed and undoubtedly the most widely used cloning vector created by Bolivar and Rodriguez. So this is the name of the scientist. This is the name of the scientist. Don't forget, my dear. Okay. The two scientists there are responsible for the construction of the, which one, the construction. Okay. Construction means, you know what about it? They built undoubtedly the most widely used cloning vector created by Bolivar and also Rodriguez. It was created in 1977. Sometimes they ask the question, the artificial plasmids also synthesized by Bolivar and Rodriguez in the year, which one, 1977. And sometimes in the laboratory of the Herbert Boyer. So this is also very, very important. Which lab? Sometimes they were asked the question. Which lab? Herbert Boyer. Okay. At the University of the California, San Francisco. Sometimes very rare, they ask the place also. You should note it down here. The first artificial cloning method, BPR 322, first. Second things, who discovered? Bolivar and Rodriguez. Okay. Third one is year 1977. Okay. Fourth one is where, exact location. That is very one. Herbert Boyer at the University of California in San Francisco. So these are the four questions in this part. The P stand for plasmids. So simply they may call as which one, sometimes they were asked the questions. What is by P? Okay, denoted as like this. The P stands for plasmids and B are for Bolivar and Rodriguez. Researcher who constructed. Okay, so here, what is that? This is the expansion. Okay, so you know what about this part, they have given the expansion here. The B denoted as plasmids. The B denoted as Bolivar. The R denoted as Rodriguez. Don't forget, here, this is the number. So this person, they may construct how many numbers of the plasmids, 322. Okay, we may construct here newly. Okay, see that one. Likewise, see the one. So, okay, what is the meaning of B? What is the meaning of BR? This is the number. Okay, how it will be? B denoted as plasmids, isn't it? The BR denoted as, okay, the BR denoted as V. So N denoted as a number of plasmids. These 3 digit, numbers. Okay, don't forget. So this is very, very important. Okay, so why they have given as a name is called as, what is that? This number, how it will becomes, this number. Okay, so don't forget, this is the expansion. The B is denoted as plasmid, the B is denoted as Bolivia or denoted as Rogers. This 22 is the number of plasmids. Number of plasmids. Okay, don't forget, my dear. This is very, very important. Okay, they may ask the question. Now you are you clear now? Okay, see that one. It contains 2. Okay, it contains 2. What is that? TWO, isn't it? TWO. 2 antibiotic resistant genes. Don't forget. Sometimes they may ask the question, how many antibiotic gene present, resistant gene present in the BP3? So one is called as which one, the ampicillin resistant. That is simply called as AMP resistant, denoted as R. And the other is tetracycline resistant. That is simply called as TET-R genes. So it is very, very important, my students. Okay. See the structure, you can easily identify. See the structure, you can identify this. This is the entire structure of the, which one in a BPR 322. Here, how many genes they told? Two genes have been reported. One is called AMPR and tetra-R. So these are the two genes present. Okay. Here they may told PS1, PUV, EcoRI, CLA, HIND-D3, BAM, H1, SAL1, DROP1, PUV. So these are all no need. There's two only you must know. The remaining things we'll see the next classes. Okay. Still now we will note it down. So these are the things. So we will see the next thing on the see here. This is the structure. Next one is also same, similar to this one. AMPR resistant gene, how many genes is present in it? Okay, in the BPR. There are two genes. What are they? One is called as ampicillin resistant gene. That's why they just named as a AMP resistant gene. And a tetracycline resistant gene that is called as tetra-R genes. Both are present in it. Okay. So next one is, see that structure is the same. Where it discovered 1977, Rodriguez, BPR 322. The P denotes as plasmid, BR denotes Bolivar and Rodriguez. The 322 number of distinct users. So I told you simply numbers only, but here there are number of distinct users. See that BPR 325, already told you here 22, 25, 27, etc. So it also extended. Okay. So this is the meaning of the BPR. Okay, that is also very, very important. So next class, we will see the remaining things on the next classes. Today, I will, what is that? Summarize all those things. Okay. See that what are the things is there already we discussed. Anyhow, see this is the one. Vector-mediated gene transfer. First of all, a cloning vector. Okay. The cloning, how it will be constructed. You know, there are two types of DNA molecules, very, very essential. One is called as the important elements is the recombinant DNA technology, or the gene cloning is the vector. The random DNA or cDNA segments, and specific DNA is linked to the small replicating DNA molecules called as rDNA, that is called as recombinant DNA. Here what happened, there are different types of cloning vectors for use with the different types of host cell is also very, very important. According to their closed cell, according to the vectors also they are used. Okay. So the largest number exists for the E. coli, that is very, very important, particularly the plasmids. The plasmids, how it will be, not only the plasmid, other components also, other microorganisms is also essential for the vectoring, isolation of the vector process. Besides, there are cosmate pages and page mits and yeast artificial chromosomes and transpons, bacterial artificial chromosomes, etc. These are all considered getting known as vector. Particularly E. coli is the most important one. Choice of the vector already know, but the vectors is an atomously replicating inside the host cell DNA molecule designed for the plasmids or feeds DNA, carry a foreign DNA inside the host cell. The vectors should have the following features, then only it will be used for the biotechnology. The must contain replicon, otherwise called as ORI, origin of replication process, origin of replication, that enable to replicate in the host cell. It's a very, very important thing. Okay, and then the several markers that were used. Okay, so then only we can easily identify which one is transformed gel, which one is not transformed gel. We can easily identify. That is called as what is the genes for the ampicillin tetracycline resistance genes. Already we discussed it. And not only that, and also it's have a unique cleavage sites also. Then only the marker gene as well as the insertion of the foreign DNA into the marker gene lead to the inactivation of the and identification of the recombinant DNA molecules. That things we can easily do it. Okay. So next, for the process during the when we consider as the process, the DNA should contain suitable control elements such as the promoters, terminators, and ribosome binding sites. Also very, very essential one. And preferably a small size. The vector should be how much? It will be there, you know. So particularly equal to the 10 kb. Kilobase is the transformation, transduction, and the electroporation is also very, very important. This process also they will do. Then only come to the conclusion. Okay. So stability of the host, stability in the host. The vector should be stable in the host cell after infection. It should not get the lost in subsequent generation. This permit replication of the vector producing larger copies of the gene interested. Okay. The ability to control their own replication. This is also very, very important. High number. Then only we will get the high number. When we will stop, it should be stop. Otherwise, what happened? It will be very complicated. The multiple cloning site is also very, very important one. Okay. And the next should not be transferred by conjugation. Naturally, when the both positive and negative bacteria come in contact close together, there is that combination. The sexual transformation, what happened? The conjugation canal is also formed. So that time, what happened? The transformation of the genes from the one bacteria to another bacteria. It will not happen. Okay, normally the conjugation it will happen. So such things it should not be transferred by the conjugation process. Such type of bacteria is also very, very important. Examples of vector plasmids. Plasmids are double standard closed circular DNA molecule, which is very, very important, which exists in the cell extra chromosomal unit. The size from about 1 kb to over 250 kb. Okay. And the next, based on their origin source of the plasmid, they have been divided into two major classes. One is called as natural plasmids, another one is called as artificial plasmids. Natural plasmid, example, CoL-E1, mostly present in the prokaryote and also eukaryotes. Okay. The artificial plasmid, don't forget, my dear student, this is very, very important one. So mostly the BPR 322 is the artificial one, the first discovered or invented in laboratory, in vitro. Okay. How it will be known? The naturally occurring plasmid have several limitations. For example, some are straightened and are not relaxed. Some have poor marker genes and some are too large. Okay. To overcome the limitation natural vectors, artificial plasmid is designed by the combining different elements from diverse sources. They are constructed in vitro by recombination selected segments of two or more other plasmids, natural or artificial. For example, BP or 322, the important one. Okay. The artificial plasmid vector are classified into two broad types based on their usage. Cloning vector, the expression vector. Cloning vector means what happened, it increased the number of copies of the cloned DNA fragments within the host. Such type of gene is called as what is that? Vector is called as cloning vector. Expression vector means it expresses itself in the form of protein molecule. It is used for the expression of foreign gene into a protein. That is very, very important, my dear students. Okay. So these things already we discussed. The examples of the artificial vector plasmids already know what this is the first artificial cloned vector who discovered Bolivar and also Rodriguez. It was created in the 1977 in the laboratory Herbert Boyer in California and San Francisco. The first letter P denotes plasmid, already you know about that. Second letter BR denoted as a scientific name. See that Bolivar and also Rodriguez. And the third one is called as what is it? The name, number of differences they told. Not only the 322, and some of them are what is a 325 and 327 is also present. Is also what is that? Okay. Don't forget. Okay, this is also very, very important. Okay, dear. Thank you so much for the participation. We'll see the remaining things on the next class, my dear. Okay. Thank you, Ba. Thank you so much.
Thank you,
Bye.
sir.
Thank you, Ba. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am.